Scene script. Have you ever wondered why chemical reactions in our body happen so quickly? What is the secret catalyst behind these reactions? Well, it's time to unveil the mystery. Enter the world of enzymes, nature's tiny powerhouses, and the unsung heroes of our body's chemistry. Enzymes are specialized proteins that accelerate, or in scientific terms, catalyze, the biochemical reactions occurring in our bodies. Without them, life as we know it would grind to a halt. Imagine, if you will, a city without traffic lights. Chaos, right? Similarly, enzymes guide the traffic of molecules in our bodies, ensuring everything runs smoothly. But what makes enzymes so special? Well, their uniqueness lies in their structure. Each enzyme is tailored to catalyze a specific reaction. Like a key fits a lock, an enzyme's shape allows it to bind with its substrate, the molecule it acts upon, to facilitate the reaction. This specificity is what makes enzymes such efficient catalysts. Another fascinating aspect of enzymes is their reusability. Unlike most catalysts that are used up in the process, enzymes emerge from the reactions they catalyze unscathed, ready to facilitate another reaction. This makes them incredibly efficient, able to catalyze thousands, even millions of reactions in a single second. Now you might be thinking, with so much going on, how do enzymes manage to keep up? Well, that's where the concept of enzyme kinetics comes into play. Enzymes don't just speed up reactions, they regulate them. They respond to changes in their environment, adjusting their activity to maintain the delicate balance of biochemical reactions in our bodies. But enzymes are more than just catalysts. They are also involved in a wide array of biological activities, from digestion and energy production to gene expression and cell signaling. In short, they are the conductors orchestrating the symphony of life. So enzymes are the unsung heroes, quietly catalyzing reactions within our bodies. But how do they do it? Well, stay tuned as we delve deeper into the enigma of enzymes in the next segment. Enzymes are not a one-size-fits-all catalyst. They come in various types and have different components. Intrigued? Let's delve deeper. Just as there is a vast array of chemical reactions in the biological world, there is also a diverse range of enzymes, each specialized to catalyze a specific type of reaction. Enzymes are typically classified into 6 main categories based on the reactions they catalyze: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Each group has its own unique role in driving crucial biochemical reactions, from oxidation reduction processes to the rearrangement of molecular structures. Now let's talk about coenzymes and cofactors, 2 critical components that often work in concert with enzymes. Coenzymes are organic molecules that bind to the enzyme, assisting in the transfer of functional groups from one molecule to another during the reaction. They are like the co-pilots in an enzyme's flight, helping to navigate the complex biochemical airspace. On the other hand, cofactors can be either organic or inorganic substances that help stabilize the enzyme structure or contribute to the enzyme's function. They are the unsung heroes, the backstage crew that ensures the spotlighted enzyme performs its best. Some cofactors, such as metal ions, bind to the enzyme and help catalyze the reaction by stabilizing charged particles. Other cofactors, known as prosthetic groups, bind tightly to the enzyme and participate directly in the reaction, like the gears in a well-oiled machine. But it's not just about the coenzymes and cofactors. The enzyme itself is a complex 3-dimensional protein structure with an active site where substrates bind and the reaction takes place. Think of the active site as the stage where the biochemical drama unfolds. Now that we understand the different types of enzymes and their components, you might wonder, where exactly does the magic happen? There's a special place within enzymes where they interact with other molecules. It's called the active site. Curious how this works? Imagine enzymes as the world's tiniest workers with the biggest impact. These minuscule proteins are the catalysts in countless biological reactions, and the key to their functionality lies in an area known as the active site. This is the region on an enzyme where the substrate, the molecule upon which the enzyme acts, binds. It's a precise fit, like a lock, and its key, ensuring the enzyme can carry out its task effectively. Now let's dive into the mechanism of enzyme actions. It's a 2-step process. First, the enzyme and the substrate bind together at the active site, forming what we call an enzyme-substrate complex. This is where the magic happens. The enzyme facilitates the transformation of the substrate into a different molecule known as the product. Once the product is formed, it's released from the active site, leaving the enzyme ready for its next task. The efficiency of this process is quantified using the Michaelis-Menten equation. This mathematical model helps us understand how enzymes respond to varying substrate concentrations. It's a bit like understanding the speed limit of our enzyme workers. To visualize this equation, we use the double reciprocal plot, or Lineweaver-Burke plot. This graphical representation helps us interpret enzyme kinetics, the study of the rates at which these biochemical reactions occur. It's essentially a roadmap of how fast our enzyme workers can get their jobs done. Enzymes are fascinating, aren't they? They're so small, yet they drive the most crucial processes within our bodies. And their efficiency and specificity largely come down to the active site and the intricate process that unfolds there. This is just the tip of the iceberg when it comes to understanding how enzymes work. There's more to it, like what affects their activity. Scene script. Enzymes might appear invincible, but certain factors can affect their activity. Let's find out how. Just like any superhero, enzymes too have their kryptonite. They are sensitive to changes in temperature, pH, enzyme concentration, substrate concentration, and the presence of any inhibitors or activators. Temperature affects an enzyme's activity by influencing the speed of molecular movement. As the temperature rises, enzyme and substrate molecules collide more frequently, increasing the rate of reaction. But beware, too much heat can denature the enzyme, causing it to lose its shape and function. Next up, pH. Enzymes are pretty picky when it comes to pH. Each enzyme has an optimal pH value at which it functions best. Deviate from this, and the enzyme can become denatured, rendering it inactive. Now, let's talk about concentration. Increasing the concentration of either the enzyme or the substrate can speed up the reaction up to a point. Eventually, the substrate molecules will saturate all of the enzyme's active sites, and the reaction rate will plateau. But what happens when a sneaky molecule interferes with our enzyme superhero? This is where enzyme inhibition comes into play. Inhibitors are molecules that bind to enzymes and decrease their activity. There are three main types of inhibitors: reversible, irreversible, and allosteric. Reversible inhibitors bind to enzymes with non-covalent interactions, meaning they can easily detach and allow the enzyme to function again. Irreversible inhibitors, on the other hand, mean business. They bind permanently, blocking the enzyme's active site and shutting down its function for good. Lastly, allosteric inhibitors don't play by the rules. Instead of blocking the active site, they bind to a different part of the enzyme, changing its shape and making it less effective. Enzymes are sensitive, aren't they? But did you know they also have numerous applications? Scene script. Enzymes are not just biological catalysts. They have a multitude of applications. Want to know more? Let's dive right in. Picture this. You're enjoying a loaf of bread. What you might not realize is that enzymes played a critical role in its creation. In baking, enzymes like amylases and proteases help break down starch and proteins during dough fermentation, resulting in a fluffy and delicious loaf. But the magic of enzymes doesn't stop at the bakery. They're also pivotal in the brewing industry. Enzymes are used to break down the complex sugars in grains, enabling yeast to ferment them into alcohol. In the realm of medicine, enzymes are nothing short of lifesavers. They're used in diagnostics, for example, to measure enzyme levels in blood tests as an indicator of certain diseases. Therapeutic enzymes are used to treat conditions like digestive disorders and even cancer. L-asparaginase, an enzyme, is used in the treatment of acute lymphoblastic leukemia. In the field of biotechnology, enzymes are used in genetic engineering to cut, replicate, and attach DNA as needed. This allows for advancements in medicine, agriculture, and more. And let's not forget about the environment. Enzymes are used in bioremediation, which is a process that uses organisms to clean up pollutants. Certain enzymes can help break down harmful substances, making them less damaging to the environment. So, you see, enzymes are everywhere, silently working behind the scenes, making our lives better in countless ways. To wrap up, enzymes are much more than just biological catalysts. They're the secret ingredient in your favorite loaf of bread, the silent helper in creating your favorite brew, the unsung hero in medical breakthroughs, the ingenious tool in biotechnology, and the silent warriors protecting our environment. Enzymes, The unsung heroes of our bodies are more complex and versatile than they appear. Subscribe for more because you won't find me later.